Cellular respiration and fermentation are both processes that break down organic molecules in order to yield some energy for the cell. However, there are some notable differences between the two as well. So what we want to do here is we want to compare and contrast what's similar and what's different about cellular respiration versus fermentation. So first off, if we talk about things that are similar or the same for both of these. As we said, both are going to break organic molecules. Now, they're breaking the organic molecules. The big difference between them is the extent to which they actually break those molecules. So if we're starting with a glucose molecule, in the case of cellular respiration, we are going to completely break that molecule down. So since we're completely breaking that molecule down, we are going to yield lots of carbon dioxide, because carbon dioxide will be the end result of the breakdown or full collection of the electrons from the carbon. In the case of fermentation, it is a partial breakdown. Because it's a partial breakdown of the sugar, we will only partially collect the electrons, and that means that we sometimes, and only sometimes, will yield a little carbon dioxide. The only time we're going to yield a little bit of carbon dioxide is if we're doing the ethanol fermentation or alcohol fermentation. If we were actually talking about the lactic acid fermentation, we wouldn't be yielding any carbon dioxide at all. There is going to be a difference in how they actually make ATP. So both of them will make ATP. That is the same, but how they make it is going to be different. Cellular respiration is going to use that substrate level phosphorylation and also some chemiosmosis, and most of the ATP that's made is going to be made by the chemiosmosis process. When we're talking about fermentation, it is only going to make ATP by substrate level phosphorylation. If we talk about stages, cellular respiration uses 3 stages. We had glycolysis, the citric acid cycle or the TCA cycle, and then we also had the oxidative phosphorylation. In the case of fermentation, it is only glycolysis plus a few extra steps. And those few extra steps are going to be used to regenerate that NAD+. If we clear this off so that we can add some more onto our list. So additional things, if we talk about the yield of cellular respiration versus fermentation, per glucose that we start with, cellular respiration yields a maximum of 38 ATPs. So that's quite a lot. And when you compare that to fermentation, fermentation is a maximum of 2 ATPs. So it's a lot less efficient when we're talking about fermentation. It's actually 19 times less. Okay, so much, much less. And then another thing is that cellular respiration is aerobic versus fermentation, which is anaerobic. What those words mean is that this one requires oxygen. This one over here does not use any oxygen. And that's really the reason why the third stage of cellular respiration doesn't happen in fermentation. And then one final last thing is that if we look at the end products, as far as what happens to our organic molecule, cellular respiration is going to break it into inorganic carbon. So inorganic carbon is basically your carbon dioxide. So it will take every single carbon in that original glucose molecule and yield it eventually back to the atmosphere as carbon dioxide. In the case of fermentation, we end with organic molecules. So we have organic waste molecules. Those organic molecules, because they are organic, there is a lot of energy that's left unharvested inside of them. So that would be the ethanol, or it could be the lactate if we're talking about the lactic acid fermentation.